I'm Chris Cowell, Director of Research at Sydney Children's Hospitals Network, and wish to welcome you all to what will be a very exciting webinar this afternoon on focusing on gene therapy and its potential transformational impact for children with genetic disorders. I'd like to begin by acknowledging that we are hosting this webinar from the traditional lands of the Gadigal and Bidjigal people of the Eora Nation in the east and the Barramadigal people of the Darug Nation in the west of Sydney. We acknowledge the traditional custodians of the various lands on which we are all meeting today around this nation and potentially some overseas, and the Aboriginal and Torres Strait Islander people participating. We pay our respects to elders past, present and emerging, and celebrate the diversity of Aboriginal peoples and their ongoing cultures and connections to the lands and waters of Australia. Once again, welcome to our webinar on Advanced Therapeutics, transforming the way we care for children. A few housekeeping messages. A video of the webinar will be emailed to all attendees and available via our website in coming days. Our Q&A panel will be answering selected questions at the end of the webinar. Please submit questions through the Q&A tab in the Zoom controls. If you see a question submitted that you would also like to have answered, you can upvote that question by clicking the thumbs up beside it. We'll also be providing answers to questions via our website in the coming days after the webinar. So getting on to the webinar. We're fortunate having two outstanding speakers, Professor Ian Alexander and Associate Professor Michelle Farrar. Ian is the head of the Gene Therapy Research Unit, a joint initiative of Children's Medical Research Institute and the Children's Hospital at Westmead. He's a senior staff specialist in metabolic medicine and also Director of Laboratory Research at Kids Research. He's a professor in paediatrics and molecular medicine at the University of Sydney and an honorary consultant in clinical genetics at Westmead Hospital. His specific expertise and interests including virus-mediated gene transfer with a focus on target organs, including the liver and bone marrow, both of which show great promise for the treatment of genetic disease in children. He has greater than 25 years research experience in vector design and gene therapy. Associate Professor Michelle Farrar is a clinical academic in paediatric neurology at UNSW Sydney and specialist child neurologist at Sydney Children's Hospital, leading the neuromuscular clinical and research programs. She is a leading researcher in pediatric neuromuscular and rare neurogenetic disorders, successfully translating research to improve health practice and outcomes for patients, their families, and the community. Michelle's team focuses on developing and translating innovative ways of providing the best possible care from a time of diagnosis through treatment and beyond to people with neuromuscular and neurodevelopmental diseases. I welcome Ian, who will commence this webinar with an overview of gene therapy, and that will be followed by Michelle and then the Q&A session. Ian?
So thank you, Chris. And so my brief today is to give a brief introductory overview of the gene therapy field in advance of Michelle's story, sharing the SMA story. So I've been in the medical profession and biomedical research profession for approaching 40 years, and I have to say that I have never been more excited than I am right now about the transformative power of modern genomics for healthcare, not only for therapeutics, but also for diagnosis and prevention. But today, I'm focusing on gene therapy, which I regard as one of the major therapeutic paradigms that's going to drive modern genomics forward. So what is gene therapy? Well, put simply, it's the use of genes as medicine, but I thought I'd start by sharing a more detailed definition and capture some critical concepts. So it's the transfer of DNA or RNA or synthetic analogues thereof into the somatic cells of a patient to repair a genetic defect or to introduce a novel function for the purpose of curing or alleviating disease. And I just want to highlight a few points there. Somatic cells, in other words, not germ cells. So any genetic modification that we make is restricted to the patient that's treated, is not heritable, and doesn't affect future generations. Secondly, it not only has the capacity to repair genetic defects, but we can actually confer novel function. And the CAR T cell field, and I'll mention that later, is an example of that. And finally, gene therapy is not about helping us run faster, jump higher, look more beautiful, or be more intelligent, so-called advancement or therapy. It's about alleviating or curing disease. So it's very simple in theory, but it's amazingly complex in practice. So I thought I'd begin by touching on the scope of gene therapy. So when gene therapy was initially conceived, most people thought only of genetic disease, but as time has passed, it's become increasingly apparent that gene therapy can be applied across a much broader set of target conditions. So cancer, infectious disease, vaccination, and in fact, several of the vaccines being developed for COVID-19 are gene-based vaccines. And the bottom line is that any biological process controlled by the function of genes is a potential target for gene therapy. So really, we're only limited by imaginations. The other introductory point I'd like to make is that gene therapy is pervasive. If you go to the literature and you look across all of the different subspecialties that where gene therapy research is going on, they're virtually all represented. So this is relevant to virtually everybody in the medical profession. So what I'd like to do now is just put advancements in some sort of historical context, because it's very easy to take for granted the immense amount of genetic knowledge that we currently have. But the truth is, most of that's accrued relatively recently, and certainly mostly since the Second World War. So I've just got a timeline here dating back to 1944, when Oswald Avery proved that genes were made of DNA, not proteins as previously thought. 1953, Watson and Crick defined the structure of DNA, the antiparallel double helical structure with base pairing. And then in 1961, Marshall Nirenberg and colleagues at the NIH actually cracked the genetic code. But there was a series of platform technologies that needed to be developed beyond that before the gene therapy era or attempts at gene therapy could finally dawn. And they were the development of cell and tissue culture dating from the late 50s, the recombinant DNA technology revolution dating from the early 70s, and the cloning and sequencing of human genes that began in the mid-70s and culminated with the Human Genome Project. And then the development of virus-mediated gene transfer that really began in the early 80s and was considered safe enough to attempt a gene transfer by about 1989. So the first gene transfer trials in man, again, these took place at the National Institutes of Health in the US. The first in 1989 was actually a gene marking trial where Steve Rosenberg took tumor infiltrating lymphocytes from patient tumors, genetically marked them with a retrovirus vector, and then returned them to the patient. So soon after that was the actual first trial with therapeutic intent. And this was an attempt to treat infants with a form of severe combined immune deficiency caused by adenosine deaminase deficiency. And Mike Blaise, French Anderson, and Ken Culver are pictured here at the press conference announcing this trial about 30 years ago. And again, they used gamma retroviral vectors and they were targeting peripheral T cells. And I've just shown Dusty Miller here, who's a former postdoctoral mentor of mine who actually developed the gamma retroviral vectors that were used in this trial. So he was what I call a tool maker, and the others, the tool users. So the first success, so that first trial that I've just mentioned didn't confer therapeutic benefit, and a decade passed before we saw the first therapeutic benefit conferred in gene therapy. And this was published in the journal Science. It was treatment of another form of severe combined immune deficiency, this time caused by gamma-C deficiency. And it was a team at the NECA Paediatric Hospital in Paris, led by Marina Cavazzana-Calvo and Alain Fischer. And this involved harvesting CD34 cells from the infant bone marrow, taking them to clean room facility, genetically introducing the gamma-C gene with a gamma retroviral vector, and then taking those modified cells back to the patients, infusing them into a peripheral vein, where they then home to the bone marrow and over a few months reconstitute the immune system. And if you look at the last line of this abstract, you see it says, thus gene therapy was able to provide full correction of disease phenotype and hence clinical benefit. So if you now map that first trial and the first success onto this histogram showing clinical trials over a 30-year period, and this number's around 2,000 trials in approximately 40 countries, the first thing that jumps out is that there were some 400 clinical trials between 1990 and the year 2000 before we saw any clinical success. And the obvious issue is then why is it so? Why did it take so long? This is Inder Verma. So Inder Verma is one of the early pioneers in the gene therapy field. He was a tool maker. His lab developed early lentiviral vectors. And he used to say in presentations that there are only 3 problems with gene therapy: delivery, delivery, and delivery. And he would go on to say it isn't going to be a problem to make gene therapy work if we have an appropriate set of tools. But without them, we don't have a hope or a prayer. So I'm just going to say a little bit about gene transfer technology before moving to more clinical focus. So there are multiple gene transfer technologies out there. I've listed several here. They're highly complex, they're rapidly evolving. They tend to be disease agnostic, but not target cell or organ agnostic. And they fall into 2 basic classes: physicochemical means of delivery, for example, nucleotide analogues and lipid nanoparticles, and genetically engineered viruses. And I'm showing the genetically engineered viruses as a bigger image here because this is the predominant system where we're seeing most of the clinical success. But the other technologies are in the clinic and growing over time. So let me just tell you a little bit about recombinant virus. How do you make a virus into a vector? So it's really quite simple, at least conceptually. You have to understand the native structure of the viral genome. You actually gut the virus, remove its own genes, pop them into a genetic element that you can use in a producer cell, and then you engineer in your recombinant viral genome that's got your therapeutic gene, or potentially a template for homology-directed repair or homology-indirected repair if we're doing a genome editing approach. And then you can trick cells into manufacturing viral particles that, instead of containing the wild-type genome, actually contain the recombinant genome. So that becomes a vector, and it's slipstreaming viral evolution. It has all the gene transfer properties of a virus, but it's not able to infect or cause disease. It's a simple vehicle for carrying DNA or RNA into cells, depending on the type of virus that the vector's derived from. Now, Inder wasn't quite right when he said there's only 3 problems. I think there's actually 4. And one of the other reasons that there was quite a period of time passed before we saw successes was that I don't think that they were bringing together the right levels of expertise of tool users and tool makers to define target diseases. And what I've tried to capture here in this Venn diagram is domains of knowledge. There's people who have detailed knowledge about gene delivery and editing technology, and there tends to be another group of people who have a lot of knowledge about the target disease, its clinical presentation and management, and you really need to get those people together to choose your target disease. And what I'm attempting to show in the middle here is only a small subset of the diseases that are potentially amenable to gene therapy are currently within reach of technology. So if you don't go after a disease in this space, you're doomed to failure. If you want to go after a disease in this space, then you've got to push the technology along before it will be ready. So a critical thing is choosing the right target and knowing it's within reach of the contemporary technologies, and that requires a multidisciplinary team. So coming back to this histogram, I just wanted to plot some really stunning successes, high-profile successes onto this, and the consequence of that. So really, after that first success for SCIDX1, there was still almost another decade passed before we started to see some stunning successes. And this one is the treatment of haemophilia B in adult patients using a recombinant viral vector. And there was a follow-up paper in 2014 reporting that many of these adult patients became factor independent. And that was one of the major stimulants in the field dating from about 2015. In 2012, the first paper describing the use of chimeric antigen receptor modified T cells, the CAR T cell field, was born with this publication in the New England Journal of Medicine. And that's also grown into a massive field. And again, viral vectors involved, this time by ex vivo gene transfer. And then finally, in 2017, we saw the publication in the New England Journal of the single-dose gene replacement therapy for spinal muscular atrophy. And Michelle is going to tell us much more about that story. But the net effect of these developments and a number of others was that it caused an upsurge in interest in the gene therapy field, particularly from the commercial sector. And this target diagram attempts to capture that this activity. So this is due diligence by Wells Fargo looking to invest in the gene therapy field. All of these little mosquitoes on this target are actually therapeutic companies, mostly small biotech, but some bigger ones like Pfizer. Around the outside are the organ system areas that these companies are going after, with disease indications written next to each of the companies. And as you move from the outside to the inside of this diagram, you're going from early phase trials through late phase trials to marketed products. And there are now at least 6 marketed gene therapeutics on the planet, and many more coming through very fast. And there's another equally busy diagram showing the companies that have come into the CAR T cell space. So an absolute explosion of interest, investment that's now superimposed on top of all the academic activity in the field. So what about Australia in the global context? Well, we have to be realistic about Australia. We do about 3% of global R&D, meaning 97% happens overseas. And what's happening, of course, in the gene therapy field is many of these stunning developments are happening overseas, and they're beginning to flood into Australia. And these companies are particularly interested in Australian patient cohorts and getting access for early phase through late phase trials. And I've listed some of the therapeutics that have reached licensing that are actually already coming into Australia and already being applied: SMA, RPE65 deficiency, and CAR T cell technology for acute lymphoblastic leukemia. I emphasize that all these are virus-based, and I put some big US dollar numbers here to help people appreciate the type of cost associated with these. But really what we want to do is see more therapeutic development in Australia, and there are a number of barriers or obstacles along the therapeutic development pathway that we're sequentially knocking over. We've been putting a lot of effort into health system readiness, but there's also issues related to access to gene transfer formulations at clinical standard. And that's one thing I do want to just mention briefly, that here at Westmead, we're developing an advanced viral manufacturing capacity for Australia. And we hope that this will be up and running within the next 12 months or so. And we're really taking a global vision here. So the future of gene therapy is very bright. And I just wanted to finish by mentioning the rapidly evolving field of genome editing. Up to the present time, most of our interventions have been simple gene addition, but it's moving in the direction of using gene repair. Many will have heard about user-targeted nucleases and base editors. These are really emerging rapidly, and we're starting to see clinical trials in this space. And this is profoundly exciting. This is being able to go into the human genome in vivo to define target cells and edit the genome at single nucleotide resolution. So that's the exciting trajectory of the field. And I just want to finish before handing over to Michelle by saying, really, we are just at the beginning of the field. And if all of the theoretically amenable diseases in gene therapy were fruit on the tree that I've diagrammed here, then the reality is that all of the contemporary successes are relatively low-hanging fruit. And as we reach further up the tree, we're going to need to push the technological progress along to get the highest hanging fruit. So it's the beginning of a profoundly exciting journey. And I think we're going to see an absolute avalanche or tsunami of gene therapy flooding into Australia. And with that, I'll stop sharing my screen and hand over to Michelle Farrar. Thanks very much for listening.
Thanks, Ian, and hello, everybody, and thanks for joining, and thanks to Kids Research for asking me to present today. So I think, as Ian has said, the future has a number of possibilities which are very exciting, and the big questions are which disease, which patient. And I'm hoping by sharing the SMA story that this really provides an example of clinical trial readiness and the path forward for a number of diseases. So as a child neurologist, I've got many memories of diagnosing children and their families with spinal muscular atrophy or Wounding-Hoffmann disease, and informing parents that there was nothing that I could do to save their child's life. And Advanced Therapeutics bring hope. And in this webinar today, I'm going to share our experiences at Sydney Children's Hospital Network of implementing clinical trials of advanced therapeutics in spinal muscular atrophy, and really describe how we are moving towards a treatment paradigm from palliative care to reactive care, and in the future proactive care, and what the challenges are for clinical translation. So for those of you that don't know, spinal muscular atrophy is a motor neuron disease. It causes floppiness, progressive weakness, and loss of motor function. The disease has a range of severity, and the most common and the most severe form is type 1, and that's 60% of infants. And 8% of those affected would survive beyond the age of 20 months. And really, it was the scientific characteristics of the disease that really led to it being a paradigm to move forward for therapeutic development, that the gene was understood, the phenotype was understood, and it has an incidence of 1 in 10,000 children. And without a disease-modifying therapy, SMA was the most common genetic cause of infant death. So to move towards clinical trials, the natural history was critical. And this is work that we did before there were any disease-modifying therapies of the Sydney Children's Hospital cohort of SMA of 70 patients. And it really describes the natural history of what I mentioned of being a very severe leading cause of infant death. And it was understanding the genetic underpinnings of SMA that was critical to driving therapeutic development. So SMA is due to a non-functional survival motor neuron 1 gene, and that entails deficiency of the survival motor neuron protein. There's an almost identical backup gene, SMN2, and it's able to make a small amount of protein. However, there's a single nucleotide difference between in exon 7, a C to T change, which doesn't actually change the amino acids, but changes the way splicing is undertaken in exon 7, particularly in SMN2, so that the majority of transcripts lack exon 7 and they're unstable. And so I think it was when I started my PhD in 2009 that the director of the NIH at the time said, this is the neurodegenerative disease that's closest to treatment. And it was really understanding the genetic underpinnings of it. And the therapeutic approaches were twofold. Firstly, focusing on SMN2 using antisense oligonucleotides to enhance the inclusion of exon 7 in mRNA transcripts. And that drug is called nusinersen, and it binds to an intron splicing silencer segment. It thereby suppresses splicing silencing, and this results in increased proportion of full-length SMN to mRNA and full-length protein. And it was after a decade of preclinical research and promising safety data that it was ready for this to launch into clinical trials. And the second approach was replacement or addition of the SMN1 gene. So going back to the first approach of SMN2, after the preclinical studies and some early phase clinical studies, it was in 2014 that we at Sydney Children's Hospital enrolled our first infant with SMA type 1 in a phase 3 randomised, double-blinded, placebo-controlled trial, and it was called ENDEAR. So this is a photograph of us administering the medication in one of our patients. It's administered by intrathecal injections. There's 4 doses in the first 2 months, and it's followed by maintenance injections every 4 months. And it's thought that it would need to continue through life, but we don't really know the answer to that. In 2016, following an early analysis, the trial was stopped. It was due to a futility of continuance, and that was because the primary endpoint was met and it was felt that it was unethical to continue, and all patients moved over to the medication. And really the endpoint was that it demonstrated significant increases in survival. And consequently, in December 2016, nusinersen was the first approved treatment for spinal muscular atrophy in the US by the FDA. And as such, we were able to start treating the first patients with SMA type 1 in Australia before TGA approval with an expanded access program. Subsequently, 2 years later, in 2018, nusinersen was listed on the PBS for people with SMA that were symptomatic, initiating therapy before the age of 18, and SMA became a treatable neurodegenerative disease. So as Ian alluded, in the same issue that the ENDEAR study was published in the New England Journal of Medicine, a similar study alongside that was also published, and it was a phase 1 study led by Professor Mendel in Ohio, and it described the results of treating 15 infantile SMA patients younger than 8 months of age with gene therapy called onasemnogene abeparvovec in a phase 1 study. And this study also demonstrated improvements in survival without the need for permanent ventilation. And this graph is one of the results from the study, and it really shows motor function on the y-axis against age on the x-axis. And what it's showing is that all SMA treatments share one common property, and that's that they work better if they're given sooner. So you can see that the children who are dosed early with higher motor function in the orange compared to those dosed early with lower motor function in the green, and those dosed late in the grey, there's a different response in motor function. So the take-home message is treating early gives better response, and that really is across all preclinical and clinical data. That motor function and baseline and age are prognostic factors for therapeutic response. So I just wanted to pause to really explain the mechanism of gene therapy for SMA. It's an in vivo therapy, and that is that it's introduced directly into the patient, and it's given through a systemic intravenous infusion. And this is in contrast to ex vivo therapy such as CAR T cell, where the cells are taken out of the patient, cultured and then given back. And so I think Ian did explain some of the mechanisms of delivering genes. So you can't just give genes by themselves straight into the body. They need to be delivered. It's like posting a letter. You need to put it in an envelope and then it gets to where it needs to be. And the breakthrough for SMA came in 2009, and this is the front cover of Nature Biotechnology. And it's actually showing the spinal cord of a rat, and the green is SMN protein throughout the spinal cord, and that has been introduced through an AAV9 vector. And the beauty of AAV9 is that it's able to be given in the vein, and it crosses from the blood into the CNS to deliver a functional copy of the SMN1 gene. And I think the other things are that AAV are non-replicating viruses, and that's okay because motor neurons, the target cells, don't divide. So once the transgene's in there, it won't be diluted through repeated rounds of cell division, and that potentially may allow for one-time dosing. So you can see the illustration on the right is that the virus binds to the cell and then it delivers the gene into the cell. It crosses through the cell, through the nucleus, and then it's uncoded and the gene is there. Now the gene sits outside your own DNA in an episome and it continues to make the protein that is needed. So just returning to the concept for SMA, that early treatment leads to the best outcomes. This really posed a critical challenge and issue for the field. If we can identify SMA pre-symptomatically and we can give gene therapy, can we transform the treatment of spinal muscular atrophy? This comes from emerging scientific evidence that the disease starts early and it must be treated early. In our clinics, we were seeing that the mean time to diagnosis from symptom onset was 5 months. It really is that the horse has bolted and you've lost 90% of your motor neurons by then. And this really revealed an unmet need in early diagnosis and treatment for SMA. Accordingly, newborn screening is important for the early diagnosis prior to or during latent motor neuron degeneration. So to take this forward, we needed collaborative experts across spinal muscular atrophy, newborn screening and gene therapy, clinical research and clinical trials to really address this question. So this was an undertaking that was very immense, as you can imagine. So the next steps included preparation and implementation of our plan. Firstly, implementation of a newborn screening pilot for SMA in New South Wales and ACT. And this was the first time that a DNA technology was used as the first tiered test in newborn screening. And Veronica Wiley superbly led the newborn screening and her whole team in lab validation and screening. And that was supported by funding from the Luminous Alliance and New South Wales Health, and statewide newborn screening commenced in August 2018. An important component looking to the future of the planning was including research implementation methodology and establishing interdisciplinary collaborations to be able to inform the assessment of the practice and the policy requirements, such that we hope that if our research was successful, it could potentially be included as a routine element of newborn screening nationally following our pilot. At the same time, we actively engaged Avexis, the commercial company making the gene therapy for SMA, and we underwent site selection and subsequently clinical trial startup. We recognised that there'd be challenges in starting up a clinical trial using a new technology of gene therapy, and we were also competing globally for recruitment. So knowing this, following site selection, the Sydney Children's Hospital team formed a dedicated working group, and it encompassed the skills to work proactively and collaboratively towards a safe and timely site activation. There was input from gene therapy clinicians, scientific experts, executive and clinical researchers to keep moving forward. And I think this approach facilitated independent assessments that were critical from ethics committees, governance and the Office of Gene Technology Regulator to have all the necessary approvals in place to start the study. So as you'd appreciate, the SPRINT clinical trial is ongoing and that the results are not yet available. Sydney Children's Hospital Network were able to be a lead recruitment site globally. And I think through the study, a number of important milestones have occurred. Firstly, based on the first study of 15 patients with SMA type 1, in May 2019, the FDA approved gene therapy for the treatment of SMA in the US. And as Ian said, it has the notoriety of being the most expensive medicine in the world at $2.1 million for a single dose. Novartis have publicly announced that there's preliminary evidence that when patients are treated pre-symptomatically with gene therapy, there's achievement of early age-appropriate motor milestones and no requirements for ventilatory or swallowing support. For the patients that were dosed earlier, there's further evidence that there's ongoing benefits with durability now up to 5 years of dosing. So I think that success brought many challenges. So there are many patients who could benefit from gene therapy through the US and worldwide, but they weren't able to access it immediately following the FDA approval. And as you would know, this would draw significant attention, in part due to the public price of US $2.1 million, but also because these kids are beautiful and attract a lot of attention. So one of the issues is that the approval for children was only for kids less than 2 years. So there was pressure to access gene therapy promptly, and a number of families commenced fundraising efforts to obtain access, including Australian families, even though gene therapy is not and still is not an approved medicine in Australia. And I think this is a highly visible example of the trend that we are facing in rare diseases, and it's a challenge that we face as clinicians, society, and for government as to how we're going to respond to this in the context of limited resources. And an important component of our research and our translational vision has been examining the health practice and the health economics to look towards the future. So while gene therapy is unapproved in Australia, some of our patients have been able to access Zolgensma through a managed access program and the TGA special access scheme. And this has been facilitated from our clinical trial framework and the fact that we had the infrastructure and the approvals in place. Our approach included detailed discussions with families to set realistic expectations. And it really necessitated education amongst all stakeholders, clinicians, scientists, and families regarding gene technology to be able to ensure informed consent, consideration of risks, benefits, and alternatives. And I think that was a little bit challenging because the clinical trial had a narrow population, and I think the population that we were talking to was different. They were heavier, they were older, they had different burdens of disease. So throughout the process today, the priority has really been ensuring safety, but it also shows the value of using real-world data to further understand new medicines and also support reimbursement decisions, particularly as there is a lot of uncertainty as the data's early and the data encompasses small patient numbers. I'd also like to add my experience of how complex the administrative requirements were and how time-consuming they were to really access unregistered medications. This included tripartite legal contracts, interactions with the TGA, customs, importation, the Department of Agriculture, taxation, GST, payment, to name a few. So for any one patient to access gene therapy, there is a minimum of 7 administrative steps, and that's before you provide any clinical care. And so I think having a framework and staff to really support this in a workforce that's literate in gene therapy is going to be critical in translation and moving forward. And so I think that has involved for us training staff and also the necessity to send samples overseas for serological testing of AAV9 antibodies, and looking for the future, it would be nice to have something locally. The complex logistical requirements have been a barrier for administering therapies at other centres to date, where families have been requesting access, and we've been able to support this to really have a vision of equitable access nationally. And this photo is actually the birthday cake of one of the kids that we were able to support access and dosing of gene therapy from interstate just a week or two before her 2nd birthday, and that was the birthday cake that her family made her because they were so pleased that they were able to access it before she was too old to get it. So it's not just genes, it's the environment, and it's also the family. So the diagnosis of a serious illness has a major impact and an ongoing impact on families. So an important component of our approach is research embedded as part of clinical care. This was especially prominent with newborn screening, and it really showed me how this can enable equitable access in diagnosis and also access to care. It also showed me the importance of our multidisciplinary team, and we were able to provide precision care and support for each family. So it was different. It could be logistical support for travel from different locations, accommodation, the need for interpreters for culturally and linguistically diverse families, genetic counselling, and also psychosocial support. It was also brought to the fore with COVID-19. So I think the success of any experimental medicine is also reliant on best practice. You can't have one without the other. So for children and families with SMA, this also included nutritional care, respiratory care, physiotherapy, and occupational therapy. So there are many people to thank for the SMA story. And I think, first of all, the infants and children with SMA, their families, and they've all been so generous and participated in all the studies. A huge thank you to Veronica Wiley for her vision and her leadership, and the whole team behind her in the newborn screening service. And across Sydney Children's Hospital network in Randwick and Westmead, the neuromuscular clinics and all of the staff associated with that, who've all, without a question, just given time generously to support the care of these families for the best that we could do for them. The Clinical Research Centre, the clinical trials team for all their extra efforts and their commitment to the care of these kids. Kids Research, particularly Chris Cowell and Ian Alexander, for their support in navigating the varying pipelines and all the changes and twists and turns that happened. And also Luminous Alliance and New South Wales Health for their support through these studies. Thank you.
Thank you very much, Ian and Michelle, to really stimulating, interesting presentations. And I just echo, I think the Michelle's comments about the need for a multidisciplinary team, and there have been so many incredible contributions across Sydney, and with ministry support, pharmacy support through Peter Barclay, and finally just also acknowledge the Commonwealth Government support in enabling therapies now in a variety of different programs. So it's now it's an opportunity for question Q&A, and I know there's a few questions there, but please submit them via the Q&A function. And I'd like to introduce Professor Craig Munns, endocrinologist and Advanced Therapeutics clinician lead at Westmead, and Dr. Laura Fawcett, who is respiratory consultant, Advanced Therapeutics clinical lead at Randwick, who facilitate the questions with Ian and Michelle.
Thank you, Chris, and thank you, Ian and Michelle, for your presentation. So the first question is for Ian, and Ian, it's what is the mode, or what are the modes of administering a vector to a child?
So really, there's two main approaches. The direct in vivo approach, so as Michelle's explained, as simple as an intravenous injection, so genetically engineered virus going straight into a peripheral vein, so that's the first route. And then the second major route is what we call ex vivo, where you're targeting a population of cells that can be removed from a patient, with bone marrow cells being the classical example, where they can be taken to a clean room, genetically modified, and then the genetically modified cells return to the patient, but again, via an intravenous route. So we call that ex vivo and in vivo. There are other approaches where direct interstitial injection has been used, but predominantly it's an intravenous route directly with the gene transfer formulation or with gene modified cells.
Thank you, Ian.
So I've got a question up here for Michelle. So somebody quite keen to know about contraindications for gene therapy.
Well, I mean, I think the first one is the vector is an AAV9. So we are testing to see if patients have previously been exposed to that and what their antibody titers are. And the FDA currently specifies that the titer needs to be less than 1 in 50. And it's not known if a higher titer would be okay. But I think the basis of that is that if you've already got pre-existing immunity, it's unlikely to work. Because it will be destroyed as soon as it gets into you. The rate of immunity goes up with age. I think it's about 5% and then it can go up to about 10% in later childhood. So that's one. I also think understanding the potential side effects that can happen is also important. So there will be an immune reaction because it's 1 billion trillion viral particles that you're delivering a gene to the cells. So I think some of the serious side effects in clinical trials have been related to liver toxicity. And so I think that the trial in X-linked myotubular myopathy, as the dose got up higher and the patients got older, there were deaths. And I think it was that those boys had pre-existing liver abnormalities. So I think being well with no active infection and a good baseline liver function and full blood count is very important for safety. And certainly if there's any, the way that we treat patients through gene therapy is that they're given, well, because it's IV, they're given steroids for a minimum of 30 days and then another 30-day wean. So it's very important that people don't have active infections, particularly as they're going to be on a long course of steroids and they're also going to be immunosuppressed.
Another question here for Ian. What are the factors that influence the durability of gene therapy interventions?
It's a very good and complex question. So it really depends on which target cell populations you go after in the body. If it's a long-lived target cell population, that's a good indicator of durability, but it also depends on the mechanism by which you deliver the genetic modification to the cell. And if you make a stable genetic modification to the genome of the cell, then that's another indication of a very durable effect. And then finally, even if you've got durable cells and durable introduction of a genetic sequence, there are regulatory elements that drive gene expression. And sometimes those elements can undergo slow shutdown. But the long and the short is that all of these gene therapy protocols and the methods and reagents being used are carefully considered to try and deliver the longest possible durability of effect in the light of the particular target condition going after. But at the end of the day, and Michelle mentioned that we've got 5 years data on SMA, and there's now a decade or almost 2 decades of data on SCID-X1. Really, the answer is going to be following these kids long term. And I saw Elaine Fisher ask this question once. They said, how long before you'll know how long this is going to last? And he said 75 years, inferring that you have to, we can have our theories, but ultimately we just have to wait and see. But all of the indications are that these will be durable therapies, and I don't think we should be too anxious that we won't see long-term benefit in conditions like SMA.
Okay, well, next question up for Michelle. I've got a few people, there's a couple of questions on a similar theme, which is about the engagement of families and about families declining gene therapy and how keen people were to take part in the trials.
So the trial was very unique in terms of its inclusion criteria. To be eligible for inclusion in the gene therapy trial that I led, you had to have genetic diagnosis of spinal muscular atrophy, be aged less than 6 weeks old and have no symptoms. So it was, I think, a very unique situation where there weren't many families that were going to come forward to be eligible. I think the nature of SMA in terms of its severity was that families felt that this gave them hope. And I think that's what the decision making for many families is, that when there's not many options, hope's very important. And the only alternative for them was to wait for symptom onset and then to be able to access PBS nusinersen. So we did have very detailed discussions that were over a number of days. It was never something that was a decision in 1 hour. where benefits, alternatives were deeply discussed, and also the risks, because this is an experimental medicine and we don't know if all the risks were. So I think it's important that GCP practice and full informed consent is undertaken. And my experience of talking with these families and supporting them, because it was a very stressful time, was that you know, there was a huge spike in stress, but as soon as their children they'd made a decision and the care pathway was undertaken, they felt supported and very appreciative of that. But a lot of families consider gene therapy as a very good option. And I don't know, I think I wouldn't like to give the message that it's acceptable for all families across all diseases, because I think it's an individual patient family-based decision. And it's also dependent on the child's baseline function and what the expectations for treatment will be. And I think that really determines response.
Thank you, Michelle. There's a question here from Alan Ma for Ian. What therapies do you envisage coming up next, and how well prepared or able are we as a healthcare system to run these trials or new therapies?
It's a very good question, Alan, and maybe the second part of your question first. I think in terms of health system readiness, we are getting there. One of the big challenges is that some of these gene transfer formulations fall under the Gene Technology Act, and we have to be able to comply with the Gene Technology Act in terms of the handling and biocontainment. Despite the fact there's no real infectious risk here, they still fall under the legislations. So we need the right facilities, and commonly PC2 to handle some of these reagents, and that's not immediately available even in all tertiary centres. So that's something we're working on. But most of the other elements, I think, are really quite readily addressable in terms of health system readiness. So that's the answer to the second part of the question. In terms of other things coming down the pike, I think we're going to see lots of ocular indications. We're going to see other liver indications. I mentioned haemophilia B in my part of the presentation. There's proposals around relatively common, compared to some of the rare diseases we deal with, conditions like PKU and other metabolic liver diseases. There's other neurological indications, and there's a number of bone marrow indications. So that's the genetic rare disease space, but then we also have to remember that there's activity in cancer as well. And we're going to see a lot of trials coming in, and we've already, and licensed therapies for that matter, and we're already seeing that in the CAR T-cell space. So it's going to become quite busy, quite active, but I'm confident we can be ready, Alan.
Thanks, Ian. Question up for Michelle. Is there, the next first one is, with SMA therapy, what degree of function is achieved with the therapy? Is it just stopping the progression of the disease, or is it achieving further function?
Okay, so I think it really depends on where the patient is when they receive it. So if you've lost 90% of your motor neurons, it's not going to make them come back. But in the clinical trials of kids that were symptomatic with SMA type 1, the publication in the New England Journal of Medicine demonstrated that all of those kids were alive, which is compared to 8% at 2 years untreated. None of them were requiring significant respiratory or feeding support, which is unheard of. And those kids were gaining motor milestones. I think the majority were sitting independently and some were walking independently. And that's when they're already symptomatic. So the expectation, and I think the message from that publication, is that there is improvements in motor function. But I think, you know, each child's different. And if you are presenting quadriparetic and already on a ventilator, your expectation will be different to a child that's, you know, very early in their disease with a mild burden of disease. So I think those discussions have to be had with every patient to set expectations around those patients. But the data that's been released publicly from Novartis around the SPRINT study is that the pre-symptomatic treatment of SMA with gene therapy is enabling early achievement of age-appropriate motor milestones.
Thank you, Michelle. Maybe one here for Ian, about the timeline before homegrown gene therapy becomes available, and maybe the steps involved in achieving that.
Yeah, look, this is another difficult one. So, we do have a clinical trial that's going to start in London with colleagues there that is homegrown in that the gene transfer, the recombinant genome and the vector technology has massive Australian participation in its development. But, we've chosen to push that along in London because of they've got a fantastic developmental pathway funding system. And so that was really access to dollars and also a hospital that's been deeply involved in this for about 20 years. But, we're rapidly getting to the point where we could equally well prosecute a trial like that locally. But as I did mention towards the end of my talk, we're setting up a capacity to generate recombinant viral vectors for clinical trials in Australia. For that trial that we hope to get started in London in Q2 or Q3 next year, we had to wait nearly 2 years for vector to be manufactured at Children's Hospital Philadelphia, because that explosion in demand and interest in the gene therapy field has meant there's a global shortage of production capacity for the gene transfer formulations. And that's really become one of the biggest limiting factors, both domestically and abroad. So, I do think we're going to see, once we've addressed that, many more homegrown therapeutics coming through. And that's very exciting. And as I said, there'll be multidisciplinary teams involved in that. And I think the time between diagnosis and novel therapy is going to contract, and we'll be able to play a very significant role in that journey from identifying an amenable disease to finding a powerful therapy.
Thank you, Ian.
Time for one more question, Laura.
Okay, so I think this is directed to Michelle on the Q&A, but might be one for both to comment on, about whether there's an Australian-wide initiative to establish a framework for gene therapy. So, if Michelle wants to take that first, as it was directed to her.
I think that would be a very important thing to do. I think that this webinar is the first of the initiatives to collaborate and share learnings through Australia. And I think particularly for rare diseases, we need to work together to get the best access for all of our patients. Certainly, it's been an immense amount of work, and we need to share it and build a workforce throughout Australia that's ready. And I'd pass to Chris and Ian to comment on the initiatives that they're leading for that.
I'll let Chris finish, but I would say that at the moment we don't have a very formal framework, but we've certainly established lots of the connectivities across, all of the major centres in Australia. And because we're a relatively small community in this space, the conversations are happening, and we certainly do want to build that sort of network. So, Chris, I might let you comment beyond that and perhaps round out from there then.
Well, I think the conversations are underway amongst colleagues, nationally. Advanced therapeutics is something that we're sort of focusing significantly on as an organisation and as a state, and how to deliver. But to do that, you do need to be working, with your government partners in your own jurisdiction and Commonwealth level, and clearly nationally. And those conversations are commenced, and really these webinars will be partly addressing that, hopefully next year, with involvement of speakers from around the country, but also talking a little bit about the nitty-gritty of how to actually enable these cell and gene therapies. It is very exciting times, as Ian started, but there is a lot of work, a lot of groundwork and a framework to set up to help us deliver for our families. At that time, it's almost 2 o'clock. I wish to thank particularly Michelle and Ian. I think 2 fabulous presentations, stimulated a lot of great questions from you, the audience. And so I know we can't all clap loudly together, but I'll just say thank you so much, Ian and Michelle. Also, thanks a lot to Craig and Laura for filtering the questions and facilitating the conversations. And thank you, the audience, for all the questions that you've actually put before us. We will attempt to address them over the next few days and send some responses back to close out that circle. And also special thanks to the staff who sit behind all of this at SCHN and have enabled this presentation webinar to go ahead today. Look, it is a very exciting time. Advanced Therapeutics is rapidly evolving and promises to deliver really, I think, highly innovative and potentially very transformative therapies for children, which will have a massive impact. There is a lot of work to be done, and we look forward to hopefully you joining us next year with further webinars. We'll recommence in February and be in contact. All the best, and thank you very much.